Our next speaker is Bramblepelt. Faye is from the Department of Biological Sciences. The title of Faye's talk is Genes Help Us Understand Our World. How can we make studying them more accessible? Thank you.
Can I tell you guys the thing that made me fall in love with biology? How much of the DNA in your body do you think is genes? Genes being the things that have the instructions that help make tissues, our body, they make us who we are. Do you think maybe like 50% of DNA, 70% of DNA? It's 2%. Every single bit of what makes you who you are is just 2% of all the DNA in your body. So what's the rest of it, right? Most of it is repetitive elements. All this junk that doesn't actually contribute to making the proteins that make you who you are. Those repetitive elements have these little carbons on them that help pack them up and keep them out of the way. Those marks, called methylation marks, are clues that I think we can use. I'm testing special enzymes that work like molecular scissors. They cut DNA right at those special methylation marks and leave behind anything that's not marked. What I've done is I've taken DNA and I've treated it with that enzyme. Hopefully, fingers crossed, those non-genic regions would get chopped up really fine, but they leave behind big chunks for the genes. Then I have a problem. I don't really know what's in my sample until I sequence it. So I'm forced to sift it out. Sift out all the small, tiny bits that I think are not going to be very important and keep behind those big chunks that, fingers crossed, are going to be genes. It's kind of like panning for gold. When you pan for gold, you take this giant chunk of information and shift out all the small bits and keep behind those gold nuggets. For me, those gold nuggets are the genes. Those genes, so far, my work has been able to produce an over 300% increase in gene space coverage compared to a control. That's incredible, because when we care about genetics, to better understand how a crop field can be resilient towards change or how it can produce more fruit, when we care about something like that, we can spend $50 on one individual and only get back $2 worth of genes. That's horrible. By increasing the amount of genes we get back per sample, I'm helping make it so we can spend a lot less money to get a lot more done. And that matters to me because genes are really important. I'm hoping we can use my technique to better understand historical samples to see changes across the global scale, or it could even have implications for human genetic health and testing. But ultimately, genetics is really cool and really expensive. So making it a little bit cheaper helps make it more accessible to all kinds of scientists, which is why I think my work is going to help change the world. Thank you guys so much.